So I'd just like to introduce to you today my PhD project, which is looking at how plant cell wall pectins are required for freezing tolerance. And freezing stress is an ever-increasing problem as we experience a changing environment. And what we see with this is an increase in early autumn and late spring frosts that plants just aren't prepared for. So working out how to increase freezing tolerance is really necessary for food security. So what do I mean when I say freezing stress? What happens in the plant to incur damage during freezing? Well, there are several aspects that happen during freezing to damage the plant, and one of those is actually dehydration. And this can happen in a few ways. So as water freezes in the soil, the plant doesn't have access to water for metabolism and this experiences dehydration. Now this can also happen as water freezes in the plant. So as ice nucleates in intercellular spaces, water then moves from the inside of the cell to the outside of the cell, causing the plant to experience dehydration and the plasma membrane to shrink. Now, an important part of the damage that occurs from this is actually when the ice thaws and the water moves back into the cell, which can't cope with the increase in pressure, which causes the cell to burst. And this is known as expansion-induced lysis. Now, that ice nucleation in the intercellular space can also result in cell rupture, as the ice pierces the membrane and ruptures the cell. And another result of the decrease in temperature is actually protein denaturation and also the production of reactive oxygen species, which can go on to cause damage to membranes and other cell components. Now, fortunately, a lot of temperate plants, including Arabidopsis here, which is the model organism that I use throughout my research, are able to undergo a process known as cold acclimation. And during this process, some really important changes take place within the plant that allow it to deal with that freezing stress. And these changes include, but are certainly not limited to, the production of compatible solutes to protect membranes, molecular chaperones to protect proteins from denaturation, antioxidant production to protect against reactive oxygen species, changes to the plasma membrane, which is quite an important one to keep the membrane fluid during decreasing temperatures, and also changes to the plant cell wall. And it's actually this last point that I'm going to focus on for my talk, because although there is some evidence that modifications happen to the cell wall during cold acclimation, there's no clear knowledge of how that can mechanistically help the plant to survive freezing stress. So it's the aim of my project to look into that and see how the plant mechanical structure, the structure of the cell wall, can help facilitate freezing tolerance. So as you may know, the plant cell wall is integral for plant survival. It dictates the cell's shape and size. It provides structural integrity. It's involved in cell-cell adhesion, and it can also help protect against pathogens. And to this end, the structure is quite a complex matrix of polysaccharides and proteins. And the main components of the primary cell wall are cellulose fibers, which are connected via hemicellulose chains. And then in the middle lamella, there is an increase of pectins, which are long polysaccharide chains, including rhamnogalacturonan, with side chains of arabinans, galactans, and rhamnogalacturonans-I and II. And these are all made up of lots of different sugar residues. And the particular pectin that I'm interested in relation to my research is rhamnogalacturonan-II, or RG2. And you can see there's quite a complex structure here, and I'm going to come back to this in a minute. First of all, I just want to introduce you to the mutant that I've been using throughout my research, which is called sensitive to freezing 8. And as you may deduce from the name, SFR8 is sensitive to freezing temperatures. And we can show this quite nicely by taking mature Arabidopsis plants and exposing them to minus 8.5 degrees centigrade for 24 hours. And then after 7 days of recovery in normal growth conditions, we can see quite clearly that the mutant, the SFR8 plants, aren't able to survive that exposure. Whereas in the wild type and in a complemented line, we see there's still evidence of growth in those plants. Now I can also show this more quantitatively by carrying out an electrolyte leakage assay. And this is where leaf discs of mature plants are excised and exposed to temperatures of -7, -9.5, and -12 degrees centigrade. And the electrolytes that leak from the cells during that exposure, because of damage from freezing, are measured and then exposed as a percentage of the electrolytes that leak after exposure to -80 degrees centigrade. And so we get this measure of percentage electrolyte leakage. And we can see that the SFR8 mutant has a statistically significant increased electrolyte leakage from cells during freezing compared to wild type. And this is, again, reduced in the complemented line, the mutant that's got the original gene expressed in it. So what is that original gene? What is the SFR8 mutation? So in our lab, the mutation of the mutation in SFR8 was mapped to the MER1 gene. And MER1 is an enzyme that's active in the fucose biosynthetic pathway, converting GDP-D mannose to GDP-L-fucose. So what I wanted to do was to carry out some GC-MS analysis of plant cell wall extracts to see what's happening to the fucose level in the cell wall with this mutation. And what I showed was that in the mutant, the cell wall fucose level is decreased to about 30% that of wild type. And again, this is reversed in the complemented line. So we know that the mutant plants have less fucose. But what is the consequence of that in relation to the cell wall? There are several of the cell wall polysaccharides that contain fucose residues. And actually, one of the most important ones of those is rhamnogalacturonan-II, as I introduced to you earlier. So if we take a closer look at the structure of RG2 here, we can see that it's composed of a homogalacturonan backbone of galacturonic acid residues, and it has several side chains made up of different sugars, which are all in blue here just for clarity. And the majority of RG2 in the cell wall is actually dimerized via a borate ester crosslink that occurs between the two apiose residues of side chain A. Now this crosslink actually relies on the fucose residue, which is also present in side chain A. Because if you get rid of that fucose residue, then you can no longer form the borate ester crosslink. And I've shown this by carrying out some gel electrophoresis of cell wall extracts that have been silver stained to show the dimer and monomer within the cell wall. So in wild type, we can see the presence of the dimer, which is the majority of the RG2 present. But in the mutant, we get this large monomer band, which is showing us that the RG2 dimerisation in the cell wall is decreased. Now, what we can do is supplement the plants with boric acid during growth, and this is able to reverse the phenotype of the monomer that we see in the mutant. And this is because with an excess of boric acid, we're actually forcing this bond to reform. So if we know that the bond can reform if we put in extra boric acid, what happens to the freezing sensitive phenotype if we put in the excess boric acid. And so to show this, I carried out another electrolyte leakage. And you can again see the significant increase of leakage in the mutants compared to wild type. But in the mutants that have been supplemented with boric acid, we can see that this is significantly reduced. So from this, we can see that it's quite clear that it's the Rg2 dimerisation that is necessary for freezing tolerance. And when we get rid of that, that's where we see the freezing sensitivity. So I also mentioned earlier that another important aspect of freezing stress is dehydration. And often when we see a mutant that's freezing sensitive, we might also see that mutant is also desiccation sensitive. So I wanted to test out this theory on SFR8, and through quite a simple leaf drying assay, where leaves were excised from plants and left to dry and weighed every hour, what I saw was a very drastic decrease in weight, and therefore showing that SFR8 desiccates much quicker than wild type on the complemented line. Now this difference seemed to be too large to be just because of evaporation from the plant. So my hypothesis then was perhaps the loss of Rg2 dimerisation is having some kind of effect on stomata. So I measured this by looking at epidermal peels taken from mature plants, treating them with ABA, abscisic acid, which is a closure signal for stomata, and measuring their stomatal aperture. And what I found is that actually the SFR8 aperture is much larger than that of wild type with this treatment. Another way that I looked at this was to carry out some thermal imaging of plants to look in more of a live way. And you can see here that the SFR8 mutant, which is in this pattern here, are much more purple and therefore colder than the wild type plants, which are orange and warmer. And this suggests that the mutant plants aren't able to close their stomata the same as wild type, because they're carrying out that evaporative cooling that makes their leaves much more cooler. So I then wondered, is it that the stomata don't have the same movement as wild type plants, or is it that they're just stuck open? And so what I did next was to use a Lycor photosynthesis system to carry out stomatal conductance measurements across the leaf in real time. And the stimulant that I used to affect stomatal openness was CO2. So you can see that with a decrease from ambient to low CO2 levels, there is an increase in stomatal conductance, which is what we would expect as the stomata open. And with an increase to high CO2, we see that decrease again. And there is a clear difference, there seems to be a clear difference between the wild type plants and the mutant plants. But actually the thing that I'm really interested in from this is the difference in the rate of opening between wild type, which is much quicker, and SFR8, which is much slower. So to me this is saying that the loss of Rg2 dimerisation is causing a change in SFR8 stomata, and they're not able to open as quickly as wild type are in response to that stimulus. So the question quite clearly now is, how is that Rg2 dimerisation affecting guard cell dynamics? And in stomata, we know that the cell wall, the rigidity of that cell wall is important for creating the shape of the stomata to open and close them. So what I want to measure is whether the Rg2 is causing a change in stiffness that's affecting the ability of those cells to open and close as normal. And I'm going to do that by carrying out some atomic force measurements, atomic force microscopy measurements. Now also, going back to the freezing phenotype of SFR8, does a change in guard cell dynamics influence freezing damage? And we know that actually the stomata can be a point of entry for ice growth that can then result in damage within the plant. So what I want to do is check some other open stomata mutants and see if they're freezing sensitive. And finally, going back to that aspect of cold acclimation, what I'd like to know is RG2 synthesis or dimerisation upregulated during cold acclimation, which would again suggest it's directly important and necessary for that freezing tolerance. And that's something that I'm working on now. So I'd just like to thank all of our collaborators that have worked on this. The Edinburgh Cell Wall Group for all their work on the RG2, Ian, who carried out a lot of the GCMS work, and my supervisor, Heather, as well, and the SEB for inviting me to talk to you today. Thank you very much.